Perfect. Thank you very much. Hello, everyone. Thanks for joining today's webinar. I'm Ying, and it's my pleasure to present today's topic on enabling ExpressCMC service with next-generation cell line platform. So here is an overview of contents to be covered in today's talk. First, we are going to discuss the CMC development acceleration. And then I will walk you through our next generation cell line development platform and show how that accelerates our CMC timeline. So the focus of the CLD platform, including the innovations in expression vector, single clone screening, and whole cell engineering. And in the end, I will briefly introduce our newly launched ExpressCMC service. Faster IND timelines are essential to bring promising therapies to patients sooner and stay competitive in the rapidly evolving biologics landscape. Accelerating CMC development reduces the bottleneck between the discovery and clinical entry, therefore enabling earlier access to novel treatments. And also reaching IND or earlier clinical milestones will quickly signal clear progress and boost valuation to the investors. Here is an overview of the timeline for a typical CMC program, starting from a PCC to IND filing, which typically takes about up to 12 to 14 months. And once the preclinical candidate is determined and move into the CMC phase, it typically starts with the DNA synthesis with the generation of plasmid vector containing gene of interest. And then later followed by the stable cell pool screening, single clone screening, and the fed batch evaluation of selected handful of good performing single clones. So by the end of this first stage, we end up with a few candidates, typically 3 to 6 top performing clones, and move on into the second PCB stability evaluation. During this stage, we have to conduct at least 60 PDL PCB stability with the cell passage up to 60 PDL plus a 14-day fed batch evaluation of the cell culture.
Sorry, Dr. Lee. Sorry to interrupt you.
Yes.
There are a height floating window in your slides. You can hide it.
All right. Does it look okay right now?
It's okay.
All right. So let's get back. So I think we were discussing this PCB stability will take quite a long time since it includes cell passage up to 60 PDL plus a 14-day fed batch evaluation of generation 0 versus generation 60 in terms of the expression titer as well as product quality. Once stability is confirmed, we end up with the top 1 performing single clone and move on into the cell banking construction for the MCB cell. And then afterwards, a confirmational MCB stability study is typically recommended for regulatory filing purpose. So as you can see, this lengthy cell line development cycle could be a rate limiting step for a lot of CMC programs, since it takes at least 6 to 7 months to select and construct a single clone for the tox batch production, as well as future large-scale manufacturing. Therefore, the focus of this talk is to introduce our next generation cell line platform that enables faster cell line development cycle, shorten the overall CMC timeline, and reduce the development risks. On the other hand, as you can see, a significant time during CMC development is spent on the process development, which include upstream, downstream, formulation, and analytical development. Common strategies to shorten the development timeline, including leveraging established platform process for some less complex common modalities such as MAPS, or initiate early phase process development utilizing the cell pool materials and try to overlap development activities as much as possible to shorten the development timeline. So for this part, actually ProBio has launched our toolbox process development platform targeting for complex modalities, such as the common formats for bispecific antibodies and fusion proteins, and to help accelerate the development for these challenging molecules. And this topic has been covered in our toolbox webinar series and will not be further discussed in today's talk. So what are the key features, cell line features, to help accelerate CMC development? So here I just list a couple of key characteristics of these ideal cell lines. First, high productivity expression system with operation robustness, which help to reduce the process optimization requirements and could provide excellent scalability across different scales and process changes. Second is outstanding clone stability. Ideally, it should allow close to 100% selected clones past the 60 generation stability study. Third is the product quality consistency between the cell pool and single clone, which enables earlier and more predictive product characterization and make the further process development much easier. And fourth is a high throughput single clone selection system, which enables faster and more efficient screening of the top producers. And last but not least, a diverse host cell portfolio that could customize the host cells that tailor to your molecule's unique challenges and unique requirements in the product. So next, I will present our platform key technology innovations in our expression vectors, clone screening system, and host cells that enable these essential cell line features. So let's first start with the expression vector. So the expression vector plays a central role in the cell line development, as its design governs the gene integration, transcription efficiency, and product quality, therefore ultimately determining the productivity and stability of the manufacturing cell line. There are a number of key elements in expression vector, which include a promoter that drives the transcription initiation in mammalian cells. And in general, we require a strong promoter in order to maximize the expression of gene of interest, such as CMV, SV40, EF1 alpha, etc. And of course, the gene of interest, which encodes the target protein, given that's an antibody, a protein, an enzyme, etc. And typically, a codon optimization is needed before the construction of a vector to adjust the codons to match mammalian tRNA abundance to maximize the expression titer. And next is the signal peptide, which is a short N-terminal peptide sequence that directs the transport of proteins to the ER for secretion. And it directly influences the secretion efficiency and protein folding of the target molecule. Next is a selectable marker, which helps select for mammalian cells that would be able to stably express the plasmid of interest. The typical selection system, including the GS system, DHFR system, and antibiotics such as puromycin or neomycin. And last but not least, a poly-A signal ensures transcription termination and improves the mRNA stability and translation efficiency. Without an efficiently designed poly-A signal, you may risk unstable or improperly processed mRNA, which lead to lower expression levels and higher product viability. In addition to the above-mentioned elements, the transposon system is gaining increasing popularity nowadays. So what are the transposons? In short, it's a cut and paste genetic elements that could effectively remove the target DNA sequence from a delivery vector. So basically, it helps to cut the gene of interest from a delivery vector and recognize certain integration sites on the genome and paste this genome in this host cell sequence. There are a number of key advantages of using the transposon systems over traditional random integration method. First is its high efficiency. Some people call it semi-targeted integration. While the integration is still somewhat random, the transposons preferentially integrate into the transcriptionally active region, therefore increase the likelihood of strong, stable expression of the target molecule. And second is high expression. It's able to deliver multiple intact copies of gene of interest, leading to higher productivity and titers compared with random integration, which typically only integrate a single copy at much lower integration efficiency. Third is simplified workflow because it helps the stable pool to recover from the selective pressure much faster. This enables rapid generation of high-producing stable pools in just 1 to 2 weeks. And also it reduces the screening burden because a greater proportion of the generated clones are high producers, therefore reduce the number of clones that need to be screened. In this table, we listed the comparison of common transposon systems, such as 350, pgBac, and Topic2. They come with different origins and could recognize different integration sites on the genome. In terms of the cargo capacity, pgBac offers very high cargo capacity compared with other two, and also can be excised precisely without footprint residue in the genome. So next question would be ensuring long-term stability. How to ensure long-term stability in recombinant cell lines? To address this challenge, we first need to go back and ask, why does my clone lose productivity over time? So once the transgene is integrated into the genome, over the passage generation, it will slowly go through epigenetic modifications, which could lead to gene silencing and ultimately lead to titer drop. There are a few key mechanisms for epigenetic silencing. First is DNA methylation, where the methyl groups are added to the promoter of gene of interest, therefore directly preventing the transcription machinery from binding. Modification could also happen to histone, where the acetylation or methylation could happen that removes the active marks, leading to tighter DNA wrapping, or repressive marks are added, recruiting proteins that condense the chromatin. Either way, it prevents effective DNA transcription in the next stage. And also, sometimes it has positional effects where the integration happen near the genomic silencers or constitutive heterochromatin that leads to passive silencing of the transgene. The effective strategy to tackle those challenges, including the use of stability elements, with a few examples listed here. So the next key element in the vector, as we mentioned before, is the signal peptide. So it's a short peptide at the N terminal of nascent protein that acts as a postal code, where it could direct the protein to the secretory pathway from the ER to Golgi and extracellular space. So why it is essential for biologics production? First, as we mentioned before, it's directly related to the secretion efficiency and will impact the product titer. On the other hand, it could also impact the cell growth because inefficient secretion can lead to ER stress and unfolded protein response, therefore impair the cell growth and viability. In principle, the signal peptide should be cleaved off by the enzymes in the ER and should not be present in the final mature protein. So sometimes if this cleave off is not completely, it will result in the signal peptide residue problem. And we will go over one example in the next slide. So since the signal peptide is critical in the productivity and product quality, so we need to conduct systematic optimization on the signal peptide and come up with a library of signal peptide to be used for different projects. So in our platform, we have tested a library of naturally occurring signal peptides from various organisms to identify the most effective one for secreting a specific target protein in CHO cells. As shown in this table, we have identified a handful of natural signal peptides that are quite effective in driving the secretion of different type of antibody and proteins. On the right-hand side, it shows a typical problem of signal peptide residues. This happened to one of our platform antibody CMC project. We identified during the single clone fed batch evaluation phase, we identified that a small 3 amino acid signal peptide residue was present in both the heavy chain and light chain of the target molecule, and the present ratio was quite significant. This challenge would be very difficult to be addressed in the CMC program because it will be given a small size, it will be difficult to be removed using the downstream process without sacrificing the yield significantly. And also this problem could only be detected by LC-MS method rather than the commonly used analytical methods such as SEC or CE purity testing. To address this problem in this project, we screened different type of platform signal peptide and identified the one with optimal expression level as well as no residue signal peptide issue. With all this discussion, I'd like to introduce our ProVal third generation proprietary ProMax vector. With a lot of extensive optimization efforts conducted in past few years, we have constructed our vector system with both high expression as well as exceptional stability. So in this vector construction, we implement a tighter lift element as well as a steady hold element to both improve its expression level, particularly for challenging molecules, as well as improve the passage stability significantly. We have tested this vector for all different kinds of molecular types, including monoclonal antibody, bispecific antibodies with different formats, as well as different type of proteins. So here are some of the data that's been tested. So the average titer of our ProMax vector in combination with our in-house host cells could reach about average 6 grams per liter for MAB molecules. For bispecific antibodies, up to, around 9 grams per liter for testing molecules, and proteins around 4 to 5 grams per liter. And it also demonstrates exceptional stability through the cell 60th generation stability study. So as shown in this diagram, for all the tested projects with all the selected clones, we have tested over 10 projects, and 100% of the single clone will pass the stability test with a titer drop of less than 30% in the stability studies without selective pressure at starting from generation 0. Moreover, our vector also shows very excellent pore and clone consistency, as shown in this results. As you can see, the tighter expression of the selected top performing pores versus the top performing clones are quite consistent, as well as the purity profile, including the SEC, CENR, CER, and charge variance profile. Moreover, it demonstrates quite consistent cell growth profile as well, in terms of the higher the top VCD growth curve, as well as the viability curve. It's also worth mentioning that our ProMax vector demonstrates excellent process robustness. So in this study, we tested a number of different type of molecules and the general operation parameters during the cell culture, including the temperature shift timing from day 5 to day 7, as well as the temperature shift set point from 33 to 35 degrees. As you can see from the results, under all of the temperature setting conditions, it demonstrates very consistent peak VCD harvest viability, harvest lactate level, as well as expression vector titer. And no lactate accumulation was observed for all of these conditions. Therefore, our vector system robustness could bypass the traditional process optimization, therefore reducing the process development timelines. Moving next into the discussion on the single clone screening platform. So as we all know, the high throughput monoclonal screening is one of the key technologies for improving the expression levels as well as accelerating the timelines. The traditional methods, such as limiting dilution of effects, have relatively low throughput and struggle to identify the true high expression single cells. The high throughput single cell screening with automatic system and microfluidic system enables much higher throughput and offers advantages such as gentle cell sorting and high monoclonal rate, easy to use, therefore enable faster and more accurate identification of high expression clones. On top of this, utilizing this automatic high throughput selection system, we have established our own fluorescent-based high-yield single clone screening platform in our R&D team. So in this approach, we utilize animal-free dye to label productive cells. During the cell pool stage, we perform cell pool staining with a fluorescent dye that could bind specifically to the target molecule. And afterwards, this material is put through the cell screening utilizing the microfluidic cell sorter, and the system will automatically pick up the single clones that express high fluorescent intensity. In this approach, the yield of the clones obtained is increased significantly by 30 to over 100%. And moreover, the proportion of high yield clone increased significantly, reach 50 to 100%. Therefore, our method transforms the high yield clone screening from a bottleneck into a single step signal to process workflow. So next, let's talk about the whole cell engineering. So whole cell engineering is a powerful approach to unlock higher productivity, improve the product quality, and greater process robustness. And by precisely modifying the key cellular pathways, we can control a number of features in our whole cells, including the metabolism, secretion, some anti-apoptosis, and epigenetics features, etc. Therefore, to enhance the either protein expression or control some critical quality attributes or control the growth profile. And moreover, with advent of some advanced gene editing tools like CRISPR, we now have greater opportunities to explore more creative strategies in whole cell construction. So one of the very popular example is the site-specific integration, where we need to first identify the genomic hotspots for high expression, and then putting the landing pods in our whole cells and ensure consistent transgene expression and reduce the clonal variability. And in this talk, we'll be focusing our library on different whole cells that could designed to express certain glycosylation patterns, including our acute fucosylation cell line, ADCC plus whole cells, low -5 whole cells, as well as high silation whole cells. Here is our proprietary whole cell libraries. So first, our baseline Topic1 Gen S cell line, which was obtained from ECACC and suspension adopted in our platform, utilizing animal product-free culture and chemically defined commercial media. And this whole cell demonstrates high productivity with roughly around 18 hours doubling time, and we have accumulated a quite extensive track record. Next is our Topic1 ADCC plus cell line, which was our independently developed proprietary cell line with optimized glycosylation profile, which achieves 100% afucosylation glycoforms. It will therefore enhance the ADCC effects and at the same time maintain high productivity, quite comparable with our the Topic1 gene S cell line. And both cell lines we have obtained the FDA DMF filing. The other soon to be launched new product is our Topic1 Silation Plus cell line, which is also our independently developed proprietary cell line, which demonstrates superior serum half-life and pharmacokinetics with elevated level of silation profile, and therefore enhance the therapeutic efficacy and help reduce the immunogenicity. So let's next go into details on these two cell lines. So first is our ADCC plus cell line. As shown in the image here, that's a typical fucosylation pathways in our cells. And as you can see, it involves a number of key critical pathways, including different precursors, as well as key enzymes in this pathway. So the general strategy and methods to remove or reduce fucosylation, including mutant or inactivate certain genes in the critical pathways in the fucosylation profile, in order to generate complete afucosylation antibodies. Some example including the FX knockout, Trojan, or butyl-8 gene knockout, or GFT gene mutant. Here is the performance of our in-house ADCC Plus whole cell system. As shown in this graph, our ADCC Plus whole cell demonstrate excellent PCB stability with very comparable titer from generation 0 to 60, with expression titer at 6 grams per liter. And also the purity, glycan profile, and ADCC activity are quite consistent. As you can see, in terms of the glycan profile, there was no fucosylation present in both generation 0 and generation 60. It also demonstrates very good stability of process scale-up in terms of the viability, cell growth, titer, purity. In terms of glycan profile, in all the different production scales, it demonstrates absence of fucosylation in the target antibody and pretty consistent ADCC activity across different scales. Another example is our Topic1 Silation Plus cell line, which is currently under construction and to be launched earlier next year. So in general, over the past few years, we have seen increasing number of clients requesting a specific glycan profile, such as high silation, because the low sialic acid content on glycan proteins could lead to rapid clearance from serum, leading to short half-life and increased risk of immunogenicity, and potentially reduce potency and higher dosing requirement. Therefore, we have established our whole cell engineering platform to design a desirable glycan pattern for your specific requirement. In this case, we have engineered our Topic1 whole cell line by genetic modification in the silation pathway. As shown in this slide, we have tested our modification in a number of different types of molecules, and all of these cases show a significant level of silation increase before and after modification. And at the same time, it shows that the titer of expression was not being impacted due to the modification. So in the next session, I will briefly introduce our newly launched ExpressCMC service with industry-leading development speed. So there are a number of highlights in our new service. First, we can get the top one single clone PCB cell within 2.5 months, which shortens from the previously 6 to 7 months timeline. And basically, we can leverage in this faster cell line development cycle. We can deliver the tox DS batch within 4 months, utilizing top performing clone. It's worth mentioning that there are some other platforms getting fast tox batch release utilizing cell pool materials. There are some limitations regarding this approach because first, the cell pool cannot be truly representative of the final selected clone. And at the same time, if a cell pool material is utilized in that early phase, it puts some limitations in the later phase single clone selection. Therefore, sometimes you basically give up some better performing single clone in this approach. And in general, we can achieve the GMP DS and DP release within 7 and 9 months. And if necessary, we can even speed this up further by overlapping some of these activities. And in general, this fast ExpressCMC service could enable the tox batch release utilizing top performing clone within 4 months, and possibly accelerating the overall IND filing within 8 months. Right now, since this newly launched service is now applicable to some less complex molecules such as monoclonal antibody and symmetric bispecific antibody with liquid injection and with DP concentration not falling under the high concentration formulation category. And we are confident that as we accumulate more experience in this type of service, we'll be open to a wider range of modalities in the future. So in the end, I'd also like to stress that in addition to our proprietary cell line development platform, we have also established our toolbox platform to accelerate the PD timeline. In this platform, we have collected platform examples to address, provide effective solutions for CMC challenges, and also establish our platform process for common format bispecific antibody and Fc fusion protein. If you're interested, feel free to reach out to our marketing team for our previously recorded ProBox webinar materials and our published papers. That's all for my presentation today. And thanks to everyone for attending today's talk. I'm happy to address any questions you may have. All right, I think I've seen. Let me pull out the question list first. So the first question, regarding the overview of CMC 40 months of the timeline you mentioned, ProBio completed the process from DNA synthesis to top clone in just 3 months. We know that it's actually difficult to achieve using mini-pool strategy. Yep. So basically, for this, actually there are 2 timelines I have presented in my slides. The first 14 months is a traditional kind of standard CMC timeline. In this approach, it will take about 6 to 7 months to construct the MCB. And now we have this ExpressCMC service where we are able to deliver the top performing PCB clone in just 3 months. So basically, there are a number of ways to shorten the overall CLD timeline. So first is because we know that our proprietary vector demonstrates excellent stability. So we don't need to wait for the entire almost 3 months to wait for the PCB stability results before we select the final clone. Second is we still, in this approach, we still adopt a mini-pool strategy. And as I mentioned, utilizing our new proprietary vector system, it helps the cell pools to recover faster compared with conventional approach. so that it helps speed up in the cell pool screening stage a little bit as well. And with combining a more effective platform strategy, that's how we are able to achieve top performing clone in just 3 months. So the second question, have you ever checked the reasons why the titer increased after 6 generations? That's a good question. So sometimes we do see, in a number of cases, we can see a slight increase in the titer of expression over generation. I wouldn't say it's uncommon, but I think this first demonstrates very exceptional stability of our gene integration into the whole cell, meaning that over the most generation of passage, we have not lose the integration of the target gene of interest in our genome with very effective stability enhancer elements, as I mentioned in my talk. And sometimes, in principle, we shouldn't expect even higher titer. I think this could be attributed also to some variability of the cell growth, as well as the analytical method as well. Next question, does ProBio have an IP for semi-targeted integration? Is there any IP dispute for assets licensed out to overseas comparing to traditional cell line development strategy? Under what circumstance would semi-targeted integration strategy will be preferred? First, for this semi-targeted integration, the transposon system I mentioned in particular, this is very commonly used transposon system, so we don't have any issue. And this strategy has been used in we believe in some other platform as well. So I would say this is a wild type transposon, free of IP issue. But again, for this particular transposon, we do not own a proprietary IP. For our vector, we filed our IP to protect the combination of different elements, including the transposon system, the stability enhancer, as well as the design of some other elements in our proprietary vector, but not regarding the transposon itself specifically. Is there any IP? So there's no IP issue because that's a wild type transposon widely used in the industry. Compared to the traditional cell line, nowadays, we actually would highly recommend utilizing semi-targeted integration over random integration, because we have seen in a number of projects that utilizing semi-targeted integration could significantly increase the titer of expression, especially for some challenging molecules such as bispecific antibodies. So we would highly recommend to utilize this semi-targeted integration method for your molecule. All right, that's all the questions I've seen from my end.
Okay, thank you, Dr. Li. Thank you for your answer and sharing today.